Hi students, today I am going to start Science and Technology Part 2. Science and Technology Part 2 contains mainly 10 chapters and it is related to biology, environment, microbiology and the biotechnology. So today I am going to start the chapter number 1, Heredity and Evolution. Okay, so in this chapter we are going to study about the heredity and hereditary changes, transcription, translation and the translocation, evolution, evidences of the evolution, Darwin's theory of natural selection, Lamarckism, speciation and the human evolution. Okay, before starting the lesson, just recall some questions. Which component of the cellular muscles of living organism carries hereditary characteristics? So the answer is DNA, that is the deoxyribonucleic acid is the nucleus are responsible to carry hereditary characteristics. Second one, what do we call to the process of transfer of physical and mental characteristics from parents to progeny? Progeny means a person, children or the young or offspring of animals and plants. The answer is the process is called as the heredity or the hereditary. Third one, which are the components of DNA molecule? So DNA has three types of chemical component: phosphate, a sugar called deoxyribose, and four nitrogenous bases, that is the adenine, guanine, cytosine and the thymine. So these all are the components of DNA molecule. Now let's start the first topic, heredity and the hereditary changes. The meaning of heredity is the process by which physical or mental qualities pass from parent to child. The process by which physical or mental qualities pass from parent to child. Evolution means the development of plants and animals. So first topic, heredity and hereditary changes. You know that the heredity is the transfer of biological characters from one generation to other via genes. So heredity is the transfer of biological characters from one generation to another via genes. Johann Gregor Mendel is the pioneer of the modern genetics. So the scientist Johann Gregor Mendel is the pioneer of the modern genetics. Around the period of 1886, it took a long time for him to understand the conclusions of his research about heredity. In 1901, the reasons behind the sudden changes were understood due to the mutational theory of Hugo de Vries. And in 1901, the scientist Hugo de Vries invented the mutational theory. Meanwhile, in 1902, Walter and Sutton observed the paired chromosome in the cells of grasshopper. And in 1902, the scientists Walter and Sutton observed the paired chromosomes in the cells of grasshopper. Until then, it was not known to anyone. Until then, it is not known to anyone. Research started in the direction of finding the nature of genetic material when it was proved that genes are carried via chromosomes. Through which 1944 trio of scientists, trio means the three scientists, the group of three scientists, Ostwald Avery, Maclean McGrath and Colin McLeod, proved that except viruses, all living organisms have DNA as genetic material. So these three scientists prove that except all the viruses, all living organisms have DNA as a genetic material. In 1961, the French geneticists Francois Jacob and Jacques Monod proposed a model of process of protein synthesis with the help of DNA in the bacterial cells. It helped to uncover the genetic codes hidden in DNA. Thereby, the technique of recombinant DNA technology emerged, which has vast scope in the field of genetic engineering. The science of heredity is very useful for diagnosis, treatment and prevention of hereditary disorder. So the science of heredity is very useful for treatment, diagnosis and prevention of hereditary disorder, production of hybrid varieties of animals and plants, and in industrial process in which microbes are used. Next topic is transcription, translation and the translocation. Can you tell? First one, Sketch and explain the structure of DNA. The full form of DNA is deoxyribonucleic acid and various types of RNA. So this is the structure of DNA. We already discussed there are four components: adenine, thymine, guanine and the cytosine. So this is the structure of DNA. Here there is a pairs of nitrogenous bases, that is nucleotides. Here the rails of DNA made up of sugar and phosphorus acid. The detailed study of structure of DNA in the 9th standard textbook, page number 183. So just refer the 9th standard textbook, page number 183. On that page number 183, they are given the structure of DNA and the types of RNA. Next one is the types of RNA. So RNA has basically three types: mRNA, that is the messenger RNA; tRNA, that is the transfer RNA, and the function of transfer RNA is brings amino acids to ribosomes during the translation. And the third type is rRNA. rRNA means ribosomal RNA, and the function of ribosomal RNA is RNA performs the function of protein synthesis. So let's start the topic transcription. With the help of RNA, the genes present in the form of DNA participated in the functioning of cell and thereby control the structure and the functioning of the body. So with the help of RNA, the genes present in the DNA, with the help of RNA, DNA participate in the functioning of the cell and thereby the structure and the functioning of the body. Information about protein synthesis is stored in the DNA. Information about the protein synthesis is stored in the DNA, and synthesis of appropriate proteins as per requirement is necessary for body. These proteins are synthesized by DNA through the RNA, and these proteins are synthesized by DNA through RNA. This is called as a central dogma. mRNA, that is the messenger RNA, is produced as per the sequence of nucleotides on DNA. Only one of the two strands of DNA is used in this process. So there are only two strands of DNA is used in this process. The sequence of nucleotides in messenger RNA being produced is always complementary to the DNA strand used for synthesis. Besides, there is a uracil in RNA instead of thymine. So uracil means a compound found in living tissues as a constituent of base of RNA. In DNA, it is replaced by thymine. So in DNA, RNA is present in uracil is present in RNA, but instead of thymine in DNA is replaced by thymine. This process of RNA synthesis is called as a transcription. So this process is called as a transcription. So this is the diagram of transcription. Here this is the DNA, this is the mRNA, and here there is a process, and this is the transcription. The messenger RNA form in nucleus comes in cytoplasm. It brings the coded message from DNA. The messenger RNA form in the nucleus comes in cytoplasm. It brings in the coded message from DNA. The message contains the codes for amino acid, and the message contains the codes for amino acid. The code for each amino acid consists of 3 nucleotides. It is called as a triplet codon. So the code for each amino acid consists of 3 nucleotides. Nucleotide means a nucleotide is one of the structural components of DNA and RNA. A nucleotide consists of base of one of the 4 chemicals, that is adenine, guanine, thymine and the cytosine, and a molecule of sugar and the phosphoric acid. Dr. Hargobind Khurana, a scientist of Indian origin, has made an important contribution in discovery of triplet codons. Okay, so who discovered the triplet codon? Dr. Hargobind Khurana for 20 amino acids. For this work, he has been awarded with the Nobel Prize in 1968 along with the 2 other scientists. Next topic is translation. Each messenger RNA is made up of thousands of triplet codons. So each messenger RNA is made up of thousands of triplet codons. As per the message on messenger RNA, amino acids are supplied by the tRNA. So as per the message of mRNA, the amino acids are supplied by the tRNA. For this purpose, tRNA has anticodon having complementary sequence to the codon on mRNA. This is called as a translation. So translation means each mRNA is made up of each messenger RNA is made up of thousands of triplet codons. As per the message of mRNA, amino acids are supplied by transfer RNA. For this purpose, transfer RNA has anticodon having complementary sequence to the codon on messenger RNA. This is called as a translation. The amino acids brought in by tRNA. The amino acids is brought by tRNA, that is transfer RNA, are bonded together by the peptide bonds with the help of rRNA, that is ribosomal RNA. During this process, the ribosome keeps on moving from one end of mRNA to the other end by distance of one triplet codon. So the ribosome keeps on moving from one end of messenger RNA to the other end by distance of one triplet codon. This is called as a translocation. Such many chains come together to form complex proteins. These proteins control various functions in the body of living organisms and their appearance too. So these proteins control the various functions in the body. Living organisms can produce new individuals like themselves due to the genes. So all the living organisms can produce the new individuals like themselves due to the genes. Only and some of those genes get transmitted to the next generation without any changes. And out of these, some of these genes are transmitted to the next generation without any changes. Due to this, some of the characters of parents and transmitted to their offsprings. However, sometimes sudden changes occur in those genes. Sometimes any nucleotide of the gene changes its position that causes a minor change. We think nothing but the mutation. Mutation means a change in the basic structure. Here, this is the diagram of mutation. Here there is a change in the basic structure. Some mutations may be minor, but some may be considerable. Example, mutation may be caused the genetic disorders like sickle cell anemia. This is an everlasting process, and it is one of the proof for Darwin's theory of natural selection. So this is the mutation, and in this video we are basically studied about the heredity and the hereditary changes, and then transcription, translation, and the translocation. Thank you.